First, I want to thank Dr. Pollack for kindly inviting me to attend this important conference. My interest in light-wire interaction and the application of cancer started 30 years ago. For training in laser medicine, I saw a simple demonstration that had a big impact on me. A high-powered Tumor laser was aimed with small medium laser and retorted water on the skin of the brain.
Microphone.
A brief pulse of light, of fire, and it were vaporizing. But to my surprise, the skin of the apple remained completely intact. I have been reading about the studies using low-energy lasers to induce non-thermal photochemical effects. So this experience led me to wonder about how selective light absorptions by water might be used to target non-thermally and non-invasively advanced systemic cancer and other complex diseases. A major obstacles for cancer drugs aiming at the single molecules targets to minimize non-specific toxicity, esteem that clinical response is often transitory and followed by relapse. Therefore, and even the acquisition of a new generation cancer hallmark is limited and made possible by the tumor microenvironment, which has been proposed that new cancer therapies should not aim solely at killing tumor cells, but at restoration, homeostasis, homeostasis, and microenvironment effects which, as we will discuss today, may be induced by light-water interaction. Light can stimulate or inhibit, that is modulate, cell function dependent on signal and receptor characteristics, which determine biological outcome. Laser photobiomodulation uses monochromatic or quasi-monochromatic blue-fluence light to induce biochemical, metabolic, biomechanical, and hydrodynamic effects that can control cellular and tissue energy, namely ATP, DTP, and other high-energy molecules. One conduct includes absorption by cytochrome C oxidase, CCO, a key enzyme in the respiratory electron transport chain. Laser photobiomodulation then initiates secondary cell signaling, resulting in implicit energy metabolism. Water further provide pathway for light absorption and transportation that can substitute and complement metabolic energy pathway through oxygen-dependent and oxygen-independent effects. Critical signal pathway in primary aqueous media. We all consider, and most maybe, laser increasingly been found to be a key player in numerous physiological mechanisms, including first contact event in which cells decide between survival and apoptosis. Consequently, external applied magnetic energy, light, may selectively target the organization of water to steer biological function. In the late 1980s and early 90s, our group complete animal testing and embarked on a phase 1 clinical trial in patients with advanced neoplasias using an infrared pulsed laser device, IPLD, which consists of near-infrared Triforce wave and ultrasonic carrier wave. The principles of Helsinki Declaration were followed and regulatory approval was obtained. Patients suffered from advanced solid tumor, including carcinomas and sarcoma, has exhausted standard treatments and had a life expectancy greater than or equal to 12 weeks. A paper in clinical cancer research demonstrated that IPLD was clinically safe and improved performance status and quality of life. Anti-tumor activity was found in 88.23% of patients with more than 10 years of follow-up. Previously, clinical experimental immunology published significant immune modulation. An article in Laser and Solid Medicine showed selective activation of programmed cellular death, that is, apoptosis, necrosis, and noitis, and associated cytomorphologic changes in neoplastic cells, but not in peripheral cells or tissues. T2-weighted microdensitometry results appearing in the same journal showed increased water content in tumor heterogeneity before tumor volume reduction and therapeutic anti-cancer effect, showing that changes in water acted as a predicator of tumor response, as illustrated by this figure from a patient with chondrosarcoma of the posterior fossa. Over the last 10 years, we have tried to document the structural, kinetic, and thermodynamic implication of our finding. Along the way, we have proposed detailed mechanisms that explain one basic premise. External electromagnetic energy supplementation can enhance and even substitute to endogenous ATP production to power and modulate physiologically reparative mechanisms which can help reestablish homeostasis, homeokinesis, even when the physiologic metabolic pathway has been compromised. Cellular and molecular basis for water-mediated long-range energy experimentation aiming at inducing and modulating physiologically reparative processes, including apoptosis, has been described as a part of a mechanism called photoinfrared pulsed biomodulation. The role of the water as an oscillator in the near-infrared laser photobiomodulation has also been documented, adding a more coherent description of the central effects of near-infrared light over redox centers and key transmembrane enzymes such as CCO. Recently, these ideas have been complemented and expanded by integrating the role of the quasi-crystalline exclusion zone described by Pollack as the fourth phase of the water. We have hypothesized that the exclusion zone might be guarded by laser photobiomodulation as energy reservoir, which cell may use to fuel cellular work and trigger cyclonic pathway and gene expression laser photobiomodulation would express effect via exclusion zone in a high-order biological system had not been attained. A retrospective analysis of the published data now provide evidence of photo-induced effects over exclusion zone. As we will see, born gut tissues monitored by protein-nuclear magnetic resonance, transversal relaxation times, and correlation times show significantly enhanced water structuring post-IPLD treatment. In addition, the microdensitometry of T2-weighted tumor heterogeneities from phase 1 trial in patients with advanced neoplasia and algorithm for tumor characterization also show significantly increased water structuring consistent with an effect over exclusion zone associated with histologically confirmed selective photo-induced tumor cell death. Using 90 MHz nuclear magnetic resonance spectrometer, we found that IPLD induced significant short-term molecular changes in bone reticular tissues. An PRT test was used to analyze observed difference in means T2 values and their reciprocals. After 48 hours, there was a statistical significant difference in 1/T2 value for irradiated specimens versus non-irradiated specimens and control group. And another one performed on 1/T2 values as a function of time also showed highly significant tendency to decrease only for irradiated specimens. The correlation times of the same group of rat was also significant and a range consistent with membrane effects attributable to energy transport, to biopolymers, and increased synthesis. Taken together, these experimental results are indicative of a large post-treatment entropy decrease, increased water structuring, and enhanced physiological reparative effects. This is consistent with the clinical data from the microdensitometry study of patients with advanced neoplasma treated with IPLD in the referred phase 1 trial. In that study, pre- and post-IPLD treatment T2-weighted MRIs were digitized using Perkin-Elmer microdensitometer with a resolution of 50 microns per pixel. The field of view was 150 by 150, and the matrix was composed of 256 by 256 pixels. The linear transformation was used to convert data to 256 intensity levels using the IBM HIPS software. Measurements were performed by independent blind third party, and all MRIs were obtained at most 3 days from controlled cytomorphology evaluation. Post-treatment means intensity values of tumor heterogeneities increased significance for all patients. In microdensitometry of T2-weighted MRIs, intensity values predominantly reflect water concentration per weight of tissues. Therefore, these results are highly consistent with the referred cytomorphology result showing a selective increase in tumor apoptosis. The slide shows pre and post IPLD treatment image from a patient with recurrent malignant papillary angioma. The next slide shows the T2 weighted MRI and raw microdensitometry image of a left cheek metastasis of the same patients with recurrent malignant papillary angioma. Red tones related to high water content caused by photo-induced cell death. Blue shades correspond to active tumor areas. The white divided partially separating the tumor is consistent with the loss of the adhesions due to anotis. This can also be seen in a smaller area to the right of the image. This image appeared remarkably consistent with the current understanding of the occlusion zone, in addition to the expansory effect of infrared radiation of the occlusion zone. Pollack has described the exclusion zone as declaring that ultrasound energy in the range of the IPLD carrier wave narrow exclusion zone, which experiences an stunning growth once it is moved. Hence, the low impedance of the ultrasound carrier wave not only allow the modulated infrared beam to penetrate deeply, but may also trigger with the near-infrared drive force wave a major synergic energy infusing effect over the exclusion zone. Of particular interest, the light blue color around the red tones strongly suggests areas of highly ordered outward. On the other hand, bright white areas of variable width between reds and light blue regions, especially around small islands of inactive tumor of the right, suggest areas of fluorescence associated with absorption of 270 nanometers of light. These white areas are consistent with the presence of structural matrices displaying marked charge and separation, marked charge and separation, and pH differences. The above fit well with the behavior of the corresponding curve obtained from an algorithm for tumor characterization by analysis of transversal relaxation rate, which show an increase in structured water associated to biopolymers and macromolecules. The figure shows the evolution over 2 months of 2 recurrent tumor masses from the same patients before and after IPLD treatment. The top and bottom row graph correspond to supraorbital and left cheek metastasis. On each graph, the left curve represents short T2 times, which correspond to the vertical section of the T2 exponential curve, whereas the right curve corresponds to long T2 times from the horizontal section of the same curve. Left curves show insignificantly higher amplitude and non-significant time changes, indicative of a more ordered water content associated with biopolymers at the tissue's interface 60 days post-IPLD treatment. In contrast, the right color reflects unstructured water. Water protons associated with increased diffusion consistent with selective tumor death photoinduced by IPLD. Such changes are consistent with the cytomorphologic results and the raw microdensitometry image, possibly proving photobiomodulation effects over exclusion zone water. To the best of our knowledge, this may be the first clinical demonstration of light-induced effect over exclusion zone in clinical. We conclude that, as proposed, laser photobiomodulation may increase potential energy in the exclusion zone, which can then act as rechargeable electrolytes via battery for external selective supplementation of energy demand required for cellular work, signal pathway, and gene expression in the presence of injury-induced redox potential. This further suggests that exclusion zone scrutiny may be used as a predicator of anti-cancer response before measuring tumor volume reduction. Thank you.
We have plenty of time for real interesting questions.
Thank you. Thank you for your nice clinical presentation. I was wondering, in your measurements, were you at all able to measure any light effects on blood flow? Because we have the hypothesis that light will increase capillary blood flow.
Los resultados que estamos presentando hoy
The results we're presenting today
son el producto de una revisión
are the product of a review
de un conjunto de data
of a series of data
acumulada en los últimos años.
accrued over the last years.
Y en ese conjunto de data
And in that set of data,
nosotros teníamos varias preocupaciones.
we had several concerns.
Una de ellas era demostrar
One of it was to demonstrate
si efectivamente por microdensitometría
if actually through microdensitometry
podíamos obtener una medición cuantitativa de la respuesta antitumoral
we were going to be able to quantitatively measure antitumoral response,
y no depender únicamente
not depending only
de una interpretación clínica.
of a clinical interpretation.
Circunstancialmente,
Given the circumstances,
el grupo de radiología de la Universidad de Michigan,
the radiology team from the Michigan University,
conjuntamente con otros grupos incluyendo el Reino Unido,
joined with other groups in the UK,
finales de los 90, muy en principio de los 2000,
at the end of the 90s and beginning of the 2000s,
habían desarrollado un software
had developed a software
para poder medir directamente difusión,
to be able to measure diffusion directly,
como hoy en día la empezamos a conocer en la práctica general.
as we see it today in general practice.
Más cuando nosotros hicimos nuestro estudio
However, when we performed our study
con el equipo Siemens de un Tesla
with a Siemens equipment of 1 Tesla
entre 1989 y 1992,
between 1989 and 1992,
tales programas no existían.
such programs were not available,
Y la medición de difusión incluso era
and the measurement of diffusion, in fact,
todavía lejano.
was something that was seen as
Hoy en
something
día
far.
hubiese sido relativamente muy fácil
Today, it would have been rather easy
con la misma tecnología de resonancia,
with the same resonance technology,
pero con los programas modernos
but with modern software
e incluso con una resolución de 1.5 Tesla, 3 Tesla,
and even with a resolution of 1.5 to 3
hay esas
Tesla,
mediciones a las cuales tú te refieres.
to be able to do those measurements that you're referring to, Jerry.
Anyone else? In the back.
Can I ask you for the wavelength of the laser?
I'm sorry, I can't hear you.
We got mic to you.
The wavelength of the laser.
904
904
nanómetros pulsado
pulse
a 3 MHz.
to 3 megahertz.
Someone else?
I have a
Yes.
quick. So there came out a paper recently which shows that pulsed infrared light can modulate the action potential properties of cells. Do you think that your therapy is related to, I guess, the electrical property of the cell? Are you changing the capacitance of the cell?
Definitivamente.
Definitely.
Infrarrojo es una banda muy amplia.
Infrared is a very broad band.
De hecho, comentando, trabajo con
In fact,
Jerry,
discussing some work with Jerry,
él tiene una especial demostración
he has a special demonstration
de sensibilidad
of sensitivity
de la zona de exclusión
of the exclusion zone
a cerca de 3.000 nanómetros.
at near 3,000 nanometers.
Yo trabajo con 904.
I work with 904.
¿Cómo es que habiendo una distancia tan grande,
How come having such a big
los
distance,
resultados pueden ser tan cercanos?
the results can be so close?
Bueno, porque son armónicas.
Because they're harmonic.
Y la respuesta en relación al aspecto particular
And the answer with regards to the particular aspect of the interaction of the infrared
potenciales
with membrane potentials,
hay varios artículos muy importantes.
there are several articles which are very important, and it's very well demonstrated
Que la energía del infrarrojo
that the energy of infrared
no solamente activa las enzimas
not only activates enzymes,
transmembrana,
transmembrane,
tal como citocromo oxidasa,
such as cytochrome oxidase,
sino que en un tiempo más corto,
but also in shorter times.
por ejemplo,
For example,
citocromo oxidasa requiere
cytochrome oxidase requires
en total
in total
3.1 milisegundos
3.1
para
milliseconds
completar su ciclo.
to complete its cycle.
Usted induce ese mismo efecto, transfiriendo energía
Induce that same effect, moving ions
con receptor agua.
with the water as receptor.
Eso ocurre en la escala de picosegundos.
That happens in the
Y en el peor de los casos, si hay
seconds, and in the worst case,
ya no intermolecular, sino intramolecular,
if there's excuse me. Yeah,
sí, la transferencia de energía
the transfer of energy
en agua
in water
es muy rápida.
is very fast.
La intermolecular ocurre en menos de 100 milisegundos.
Intermolecular transfer occurs in less than
Milisegundos no, femtosegundos.
in femtoseconds,
Menos de 100 femtosegundos.
less than 100 femtoseconds.
Cuando se habla de transferencia de energía
When we're talking about transfer of energy,
intramolecular,
intramolecular transfer of energy,
entonces es un rango de picosegundos.
then it changes to a picoseconds range.
Así que el infrarrojo
So the infrared
simultáneamente
simultaneously
excita enzimas oxidorreductoras
excites enzymes, oxidar production enzymes,
en tiempos de 3.1 a 3.6
in times of 3.1 to 3.6 milliseconds,
agua,
and
transferencia de iones,
and transfer of ions through